JK with Flora Funga Podcast. We are venturing to Gainesville, so that's where we're at right now, the University of Gainesville, and we are going to be sitting and standing and walking around with Matt Smith. And he is the researcher, the professor here on mycology, and so we're gonna go into his lab, sneak a little peek, and then also figure out how all of the specimens are collected, contained, preserved, and put into the Fungarium here. What you say is a Fungarium, we dive into what a Fungarium is and maybe the history as well. Come with me. I'm part of the Central Florida Mycological Society too, and we found one of those along the trail, and so I think it was Ben, he was explaining kind of what that is, and I was like, that's really interesting, how it like forms or makes it produce like a fungal leaf or something.
Yeah, it's basically
it's
gall.
Like a petal looking thing. It was interesting. It looked like a leaf, but it was made out of fungi instead of like the plant.
Oh, okay. Maybe this is something else. For Exobasidium, all the underlying tissue is plant.
Okay.
But when you look at it, you can see the fungus growing sort of between the cells, and then the hymenium, where it's making spores, is all over the outside.
Okay, so
it looks kind of fuzzy and white.
You'd see the difference? Okay.
Yeah,
not like to the naked eye?
To the naked eye, no, but like if you have a hand lens, you can look at it and you'll see it's kind of fuzzy.
Okay,
and that's because it's making spores all over the surface of the Exobasidium.
Super cool.
Yeah, I'm trying to recharacterize some of these things that were described more than 100 years ago.
Yeah.
And people they kind of don't know what the name is. You know, the fungus name is in the books, but like we don't really know what it is because no one's rediscovered it for 100 years.
Exactly.
A lot of times it was there the whole time, but no one really knew what it was.
And that's like what the Fungarium is kind of like, where there's always been, mushrooms kind of preserved in a way, kind of like a herbarium. But yeah, you were kind of letting us in on what even is a Fungarium and why should people even know about them? What's kind of the difference with herbarium, Fungarium?
It's a mushroom library. I like that. That was a good way to put it. It is basically a mushroom library. So, and it's a pretty big one, although there are several that are massive, like 1 million specimens. We're modest compared to that. And very based regionally, really focused on the Gulf Coast and Florida. Most of our most important specimens are Gulf Coast things.
Okay.
We have some other things as well that are interesting, but that's kind of the main reason that we exist, I guess. Right,
but it started as an herbarium, and then obviously you're like, well, there's so many mushrooms, maybe it should have its own little library.
Well, so yeah, I would say in the early years, fungi were treated as plants.
Yes.
So they were like weird plants that didn't get the memo. They were supposed to be green, and they were supposed to do certain things, and they didn't know.
Yes.
And so they were kind of treated like the weirdo plants.
I relate to that.
Similarly, lichens were also included always in, botanical collections. And then as awareness grew that fungi are different, in many places they were already kind of segregated in their own cabinets and stuff.
Right.
And so over time, collections have had to change. Most of our earliest collections are actually plant disease specimens. So this was the Florida Agricultural Experiment Station. So they had plants as a sort of a plant library. And that was geared a lot towards agronomy and by plants that were around here. So they would have examples. If someone needed them identified, they would have sort of these sheets. You can go back and try to figure out what you're looking at. Yeah. And a lot of the earliest collections were examples of plant disease. So they're like whole sheets with plants, and the plants are, covered by rust or something like that.
How would you preserve? I know we'll probably check this out later, but how do you preserve a fungus to be identified and
Great.
What are all the little things on the card to really
Great question. Track? Well, so I can show you that. I'll show you examples of actual specimens. Early workers all pickled their fungi. So a lot of the earliest specimens, at least for small things, were pickled. Which then you can see the shape and things like that. But it's not very practical when you start getting, hundreds of thousands of those things. First of all, it's a major fire hazard, right? They often have alcohol in them. But also it just takes up a lot of space. And for large fungi, it's not practical. And so over time, people started using dried specimens. In the early time, they also did things like try to press them, which doesn't work very well like plants.
True.
Because they are not amenable to that.
Very
Or they would permeate them with mercury and other really toxic things. Our collection doesn't have those things, but there are some of the older collections where you find that kind of stuff.
Wow.
You know, all kinds of good fumigants were used. And in fact, herbarium cabinets. I don't know if you guys have you seen old herbarium cabinets?
I've pressed samples in a college class before. I think it was, wild flora of Minnesota, and we would have to like collect specimen, ID them, and then kind of press them to be saved. But I've never done that really with fungi, so I don't really know. How would you keep the quality of the color and like the shape, and then maybe different stages? Like, would you keep one of each stage to ID?
Great question. So, people use all kinds of different practices. In an ideal world, if you have enough space and you're collecting a fungus, you would want lots of different collections of it so that you would be able to see some of that variation.
Right.
One of the problems with dried fungi is they lose some of their characters sort of no matter what you do.
Right.
Even if you're doing in the most ideal conditions, the color is going to be lost, obviously. Yep. And especially for some fungi, the odor is really distinctive, and that's lost. So we haven't figured out how to a way to do the odor part, but of course photography has come a long way. So we have a really good idea of how to, take photos in ways that preserve some of that information.
Right. Cool.
And then a big part of why we have the Fungarium, the fungus library, is so we can do microscopy.
Yes.
Because, you can capture that macroscopic image pretty well and get all that information. But a lot of the most important information is either microscopic or DNA.
Yep. Okay.
And when we go out to the herbarium, I'll show you, or the Fungarium, I'll show you guys and explain some of the issues we've had with DNA from preserving things here in the tropics.
Okay.
Or
that's
almost the tropics.
That's a good point too. And
but often just starting with what do we have, is just the very basic thing you would want to know if you study biodiversity, and we don't know.
Yeah. What's the baseline?
I mean, I'm supposed to be the state expert on fungi, and all the time I see things, I'm like, I have not collected that one before. And I've been here since 2011.
Yeah,
so, I mean, I know them better than when I got here in 2011, but there's still things that I find where I'm 100% sure that's a thing I've never collected, and probably I don't think I've seen it, but definitely haven't collected it before.
Yeah.
And, sort of as we tally numbers of fungi, they just keep going up. So.
Right. Yeah,
it's kind of impressive when you think about it.
Absolutely.
It can be challenging to find them again.
Yeah.
For sure.
Fun but maybe challenging job.
Well, you won't run out of things to do. So
Accurate.
that's one thing that I really like about studying fungi. You won't run out of questions and species to work on because there's so many.
Awesome. Well, I'm excited to check it out.
All right. So this is our culture lab. We have things sort of divided up. Because when you're working with live fungal cultures, you have to be really careful with them, and you need to not contaminate them.
Yes.
So we have a biological safety cabinet, which circulates the air through a filter. So if you're working with pure cultures of growing edible fungi or exobasidium or whatever it is that you're working on, you work on them inside of there. You can transfer your cultures and you can propagate them. And we have sort of various storage methods. These are all the things that are in the process right now. If you want to come over here,
I would love to.
You can sort of see that we have kind of a living library, and some of the fungi don't survive particularly well in this kind of scenario, but they'll survive long enough. And the beauty of that is, like, if you're working on something, you want to extract DNA and sequence a genome and things like that, then, sorry, we're going to go in the other room. Then you can do an experiment with your living culture. You can extract DNA and sequence the genome, right? All kinds of various experiments you could do when you have a living thing that you can't do.
Okay.
Not all the fungi grow in culture, so this is kind of limited to certain groups.
Okay. So what are some of those specific groups that you would work on in here?
Yeah, good question. We have worked on exobasidium. So exobasidium is a group of fungi that has a saprotrophic phase, but also can cause disease on plants. So Benton is going to be culturing tons of Exobasidium from recent fieldwork. Some of the truffle fungi that we work on grow very slowly, but they grow in culture. So that can be useful both for experiments, but also for sequencing genomes. So we have some projects where we're growing up as much tissue as possible. It's much easier to get good genomes from that kind of material. Several other mycorrhizal fungi that we've worked on, and also some wood decay fungi. We had a student from food science who was working on edible fungi to see how they tolerated iron and whether they might be able to uptake iron that people could then consume. It's kind of like early stages of that.
That's fun research.
People working on, we had a student working on bird's nest fungi, so we did a bunch of experiments and they involved cultures. So just different people in the lab have worked on a lot of different kinds of fungi.
And what has been like your past research that you've kind of been working on, or maybe stuff that, or future ideas?
Great question. I would say in my lab, we've worked on all different kinds of fungi. So some labs, I would say people are very focused. They work on one group of fungi, and there's nothing wrong with that. But there's so many fungi. One of the things that I really like about mycology is that because there's so much we don't know, you can work a little more broadly on multiple kinds of fungi. So I've worked on a lot. My favorite fungi, if I had to choose just one group to study, are the truffles. So you saw my email is Trufflesmith. So I've been studying truffles for a long time, and I sort of whenever I get any free time, if that really happens, I work on truffles. And some other people in the lab have worked on truffles as well.
So there's like a Florida truffle species, or
we have several. I would say Florida is not a hot spot for truffles. So a lot of my collections come from all over. All over the country, all over the world.
Okay, so you kind of go out also and like do some field work, collection around the world to kind of bring it back to your lab to work on.
When I can.
Okay, cool.
Yeah, when I can. So like for example, we had a long-term project in southern South America. So I got to go there and we collected a lot of truffles, and I have some collaborators from there as well. And so we've described a bunch of species from there. In fact, some new genera from there as well. So, so there's a lot to do because truffles are below ground. But unfortunately, only some of those can be grown in culture. We always are trying because it makes life easier, but many of them are challenging to grow in culture, in part because a lot of them are mycorrhizal fungi. So a lot of the mycorrhizal fungi, they need the host plant mostly, and getting them to grow in culture, you have to talk to them just right. It's like quite challenging. Yeah, exactly.
Give them the rain, the love that they need.
So they're more challenging than some other groups.
Have you tried anything with morels?
So we don't really have morels in Florida.
Exactly.
I have one collection of one morel, and it's like right from the border of Georgia, the highest points of Florida, you know, up near that 300 plus feet mark. But aside from that, as far as I know, we don't even have any other sites that have morels. So I really haven't focused on morels.
That'd be cool to like somehow, GMO it.
Well, the thing is, there are people culturing morels, but I think it's in places that are not quite so tropical. They really don't like tropical. They're really kind of cooler weather. They prefer cooler weather, or it appears that they must need a certain amount of cool weather probably to fruit. So.
Kind of like apples or something where they need like a freeze period.
Chill hours, So probably I'm guessing Florida won't be the best. It would be better to work on some other groups. For edible fungi, there's other groups that I think are maybe more Florida. Florida appropriate,
Yeah, what would you hope to see maybe edible fungi around here then?
Well, we have a lot of edible fungi, of course. I would say, as the Florida poison control contact, I'm very careful what I recommend that people eat out of the woods. Because if they do a bad job of their identification and they're eating things from the woods, then sometimes I get calls and emails, and it's rather unfortunate.
Yeah, you don't want that.
So I'm very cautious because of my role as the poison control person.
That's awesome. You have a lot of hats that you wear.
Yeah, that is. I mean, it's a great job because I get to see interesting fungi that people send to me for identification. But the other thing is I've now also seen I haven't seen any human fatalities, but I have seen some people get really, really sick from being not so careful, with the fungi that they're eating. And so I think there are some groups that you can safely eat, and they're pretty easy to distinguish.
Yes.
But there are some others where I would not recommend that amateurs consume, like things like amanitas, where we have edible species, and we have some species that will kill you. So
yep, absolutely.
it's good to be aware of those.
Yeah, and I found a lot of chanterelles around here, so that I feel like that's
chanterelles are pretty. Yeah, I feel like once you learn those, and actually we have a publication,
cool.
an extension document about kind of how to tell or chanterelles from other things. So
fun.
I can send that to you later if
absolutely.
you want. Okay, so culture lab. Yes. We don't bring dirty material in here. Let's go check out the other parts.
Get out. Oh, hey. I didn't even see you here.
Hello. So this is the molecular lab where, surprise, we do molecular biology. And so most of the equipment that you'll see is related to PCR, DNA extraction, all the things that you need to do to be able to get genetic information from fungi. We're doing in this lab. So it's also pretty clean space with the exception of this part, which is where we're doing all of our microscopy. Yes. So, well, we have some microscopy in other rooms as well, but this is kind of our main microscopy area. So we have a really nice light microscope in here.
So this is from, if you're having those specimens from the Fungarium and you're like, I need to maybe look deeper into the DNA.
Yeah, if you want to come and you were going to do some DNA extraction, you would do them here. If you're going to do some microscopy, you do it here. And so you can see the specimens up here are ones that we're actively working on. Yep. So we've got all kinds of different groups of fungi up here that various people are working on in the lab. And, here's an example of a project we're working on with a student who's in the lab this summer, who's an intern as part of a high school program that's on campus. And so he's working on this group of truffles, Stephanospora. He's working on a historical species that was put in a different genus a long time ago, and everybody forgot about it. And we collected some new specimens. And so he's like working over the microscope, doing extractions from some of the collections that we borrowed from other fungaria. Yep. So a lot of that action is happening here.
Right. So you would say that maybe historically sometimes mushrooms or fungi are placed in different families and genome or the genera. Yeah. And so now we're kind of going back and kind of reevaluating them. Like, what is the struggle with that? Like, how do you kind of change the opinion?
Sure.
On the characteristics.
So I think this is actually a good example. The one that we're working on right now. It's a good example because this fungus was named in the genus Gymnomyces. And Gymnomyces they're truffles that are related to russulas. Okay. And so now all of them have actually been moved into the genus Russula, but they look like little clods with a lot of spores inside. Okay. And originally it was named in that genus, which in that genus, those are all ectomycorrhizal fungi. Gotcha. So they need to be connected to a tree. Okay. And they're getting their energy by symbiotic association with that tree. Okay. Well, the genus that this actually belongs to is Stephanospora. Okay. And it's a saprotrophic truffle. There aren't that many of those, but there are some truffle groups that are related to saprotrophic fungi, and they seem to be getting their energy primarily from dead organic material. They don't need a connection with the host. Right. And so sometimes taxonomy is just like, oh yeah, you're just changing the name, but usually there's some deeper information there. Right. And this is a good example when we move it from Gymnomyces to Stephanospora. That will also give you knowledge about what the organism is doing, not just what the spores look like and sort of which name it is given, but the name, at least hopefully, should give you some information about what that fungus would look like under the microscope, where you would find it in the woods, and how it's getting its energy from. Right. Like what way is it getting energy? And so taxonomy is about the evolution, right? Understanding the evolution. But it's also about trying to understand what they're doing in the environment. Right. I would say for citizen scientists or amateur mycologists, it's mostly just frustrating because people are like, why are you changing the name again? You just did that, you know? And I can totally understand that. And it's really hard to keep up
there's a
with all the
meaning.
literature.
I mean, even with plants, they kind of name it to where you've discovered it maybe first. So
Right.
now you kind of know where it originated from. So now it is very confusing to kind of move it around. But if it's for the better of mycology...
Well... whether it's for the better or not, I can't say, and I can understand people's frustration with, you know, we're moving things around. But in an ideal world, at the end of the it's an ongoing process, and I guess in the end, we will hope for a better understanding of what these organisms are really doing, and to understand sort of the magnitude of the diversity for different groups. That's the goal, at least. I don't know if scientists are achieving that goal all the time, but that is the idea behind it.
A little push and pull, but it all works out, right?
Yeah, hopefully.
Cool.
So the microscope is a pretty important tool, as you know, for fungi. And I would say molecular tools have become increasingly more important, but we still do some pretty old school microscopy work in our lab.
Do you do any like spore print stuff, or you just kind of look more closer? Like, do you make any types of spore prints?
We do spore prints for sure, not for the truffles because most of them won't do a spore print. So if you cut them and you put them on a piece of paper, nothing happens because they're not shooting spores off. Not 100%. There are some truffles that still give a spore print actually, but they tend to be a weird examples. Most of them don't. Because their spores are being dispersed by animals that are eating them primarily.
Okay, so they don't really leave
They don't shoot the spores off. They're using animals. So a lot of them also have these, we talked about odors. A lot of fungi have odors. The truffles tend to have very strong odors. And that we think that is because then animals are attracted to them and know that they're ripe when the odors are just right and they can dig them up. And then, of course, they're depositing all those spores in a nice little spore packet. A little fecal pellet filled with nutrients right at the base of the tree, right? Often. And so yeah, we don't do spore prints for those, but for many other fungi that we work on in our lab, spore prints are very useful. And spore prints, they can be used sometimes to cultivate those fungi. It's also useful because a lot of times the spores that are in the spore prints are more obviously mature because you know they got shot off. So you can sort of infer those are mature spores versus when you're looking at them from a dried specimen, you'll have mature and immature mixed together sometimes.
Are truffles related to a pathogenic fungi?
Good question. So truffles have evolved like literally hundreds of times. So there are some truffles that are related to almost anything that you can think of in terms of trophic mode. Not everything. So trophic mode is where you get your energy from. But I would say there's really only one or two that seem to be related to pathogens. The vast majority of them are mycorrhizal fungi, and their closest relatives are also mycorrhizal fungi. And then the rest are saprotrophic, or doing something that seems like saprotrophic. There's some weird ones that we don't actually know what they're doing all the way. So those are really those are actually the most interesting ones because they're understudied. Sometimes they might be doing something totally different. This is one of my pet things that I'm really interested in as a mycologist broadly, not just for truffles, is that humans really like to put things in categories. Like something is a pathogen, something is a symbiont, right? Something is a saprotroph. But what if they're doing multiple things?
What if they're everything?
What if they're doing one thing, one part of their life cycle, and another thing, another part of their life cycle? And we know that that's true for some groups, but probably it's true for a lot more than we realize because we really don't know the full life cycle for a lot of them. So we've been working on some examples of that in our lab, but I think there's a lot more out there that people don't know yet. That's my guess.
That's fun.
You know, check in with me in 10 years. We'll see. These are examples of plant disease specimens that were sent to me, and part of my job is to identify all kinds of unknown specimens for the state. And so those are examples that came from the plant disease clinic. This is just a big puffball that I found in my neighborhood that I couldn't help myself. I had to collect because it was in really good shape. And then most of these other things will eventually go out and be incorporated into the Fungarium collection. Scott is visiting us He also brings material to us that's
Yeah.
interesting that he wants to put in our fungus library.
Right.
So he comes here. He's kind enough to deposit his material with us, and then it will hopefully be used by researchers of the future.
You
Right.
want to see this
Oh.
Roman aqueduct section?
Oh, that's
Yes. Can I get in here?
Yeah.
good.
So
Oh.
that's what we currently call Rhodotus palmatus in America, but it's actually a European species.
Okay.
And what we have in America is what William Alfonso Merle called Lentinula reticeps. And so me and a couple other people are moving it from Lentinula to Rhodotus as Rhodotus reticeps.
So, and I was trying to explain
That's
that's
cool.
important for a bunch of reasons.
So it's called a Roman aqueduct section, so it's
Didn't mean to get you involved in our
yeah,
whole.
it's all good. So it's like a cross section across the
Yeah.
different gills. So you can see I was like slicing it
Mm-hmm.
like that, and so the goal is to get a really thin section across the different gills, and then you can see the pilia pellets that are like the top of the cap and all its features.
Mm-hmm.
And this one actually has like multiple layers. It has a pilia pellis, suprapellis, subpellis, and then the trema, and then you can also see what's called the lamellar or hymenophoral trema, which is the cross section of the gill itself, and then all of the basidia and cystidia, the basidia holding the spores, and then cystidia, I don't think we know what they really do, right? They're
There's
just
speculation.
pretty. Yeah.
Well, they probably help maintain moisture. That's been suggested, but then also some of them have like chemical incrustations on them, so probably there's some chemical protection of the gills. I mean, that's what the speculation is. I don't really know of very many. I know of one really cool study from Asia where they put springtails in with russulas that were all the same species, and some of them they wiped away the cystidia that are on the cap, and some they didn't, and they found more. Springtails ate more of the mushroom when they wiped away the cystidia, and the inference was, well, maybe those kind of keep the springtails from feeding. But whether that's that's one mushroom and one
That's
study,
right.
so I don't really know how that applies to all fungi, but I think they show up enough. They probably have some interesting usage for the fungi, but we don't really know.
Cool. Thanks for sharing. Thanks for letting me peep. Yeah. Do springtails help spread spores for the fungi?
I would say maybe. We don't really
They're
know.
very small, but spores are really small.
So a lot of collembolans or springtails, they are many of them are fungi consumers. So they certainly eat hyphae, but it may also be possible that they spread spores as well. Like if they're feeding on spores and then they get consumed by
Right.
other animals and spread about, like it's
Right.
possible that spores are getting dispersed, but we don't really know. I would say.
Wasn't that a thing where the fungi like trap? What's the species that the fungi trap and like put up?
Nematode trapping fungi.
Okay,
Yeah.
Not springtails, nematodes. but
There is a species that I know of, for sure,
Ah.
that is known to trap springtails. It's described from a long time
Ooh.
ago. I don't think it's been found
Okay.
recently, but I mean,
not replicable.
you got to find it again, and that's hard because it's in soil. But I wouldn't be surprised if there are more that do it.
Yeah,
It's just very hard to find them.
Yeah, if there's a question about fungi, it's probably yes. You know.
Yeah, there's a lot of nematode trapping fungi for
Okay.
sure. In my class, we do an exercise where everybody brings in some soil that they gathered from wherever, and then we put them on plates. Just sprinkle some soil and add nematodes, and then you just do nothing, just wait, and then 2 weeks later you go back and look at them, and I would say more than 50% of the plates have nematode trapping
That'd
fungi.
be so fun.
It's pretty
That
cool.
sounds really fun.
But what's shocking is you don't have to go anywhere crazy. You
Yeah.
just go outside the building,
Yeah.
get soil, and
Yep.
you find them. They're everywhere.
So plate, soil, nematodes,
That's it.
and
Yeah.
then you just kind of like.
And
use
what
a
we
scope
do is
to
we use
look at-
water auger, so
Okay.
it has no nutrients. It's
Okay.
auger, so you've seen auger plates, but it's just the sort of thing for nematodes to move around on
Right.
and get killed by fungi. And
Perfect.
So you get a bunch of different kinds of morphology of those fungi.
Cool.
It's pretty cool.
Yeah, that sounds fun.
That's when you can separate the students who are going to like mycology from the ones who aren't. If you don't think it's cool to watch nematodes flopping around, getting eaten by fungi, probably I can't help you.
Like this is the peak of the...
Yeah, this is like Wild America in the Petri dish. If it doesn't get you pumped up, I can't. There's nothing I can do. So.
Yeah. Nice.
Okay. So the actual Fungarium is in here. So this is like the antechamber, let's say.
Oh, look at them cabinets. It is like a library.
Yeah, it's like a library.
Mm.
And you can feel it's nice and cool. In fact, quite cold if you stay in here for a long time.
Yeah.
And not very humid. So.
Yeah, I was gonna say, wouldn't the humidity of Florida kind of damper the specimens of some
It is a
sorts?
big problem for our collection. Yeah.
Oh.
So we have 2 dehumidifiers. So let me move some things out of the way here.
Yeah. What's the one where you're like, whoa?
Well, they're all like that.
I
Really.
like that answer.
I mean, it's like choosing one. Which one of your kids is your favorite?
Right.
It's like an impossible
Amanita.
thing. Right. So yeah, you know.
Wow.
It's an example of.
So
Let's find a good one that has types. So, the concept of types is useful. Okay, so we'll use this one. So, the way botany works, and also by extension mycology, is that when you name a species, you designate a type specimen, and the type specimen is like the model for the species. So it's like you're naming the species after that one specimen as the example.
Okay.
And so those individual specimens become really important because you know what it is because you named it, but then, maybe 50 years later, people aren't clear on what that is.
Yeah.
They're going to go back to the type specimen to try to figure that out.
Okay. And
so we have them sort of annotated in a special way so that it's relatively easy to, or let's say so they don't get lost as easily. We've annotated them with these little dots. So this is one, for example, from 1943 that was collected by Merle from here in Gainesville.
Thank goodness for Merle.
Yeah, and so...
And that's that one that we were going to...
So yeah, and he's also up here on the wall behind you.
Ah!
So William Alfonso Merle, he's up there because he has a very interesting life. He kind of has 2 parts to his mycological career. So first part of his mycological career happened in New York Botanical Garden.
Okay.
And then he had a hiatus. Some might call it a midlife crisis or whatever, where he didn't do mycology for a couple of years. And through a series of events, he ended up here in Gainesville.
Wow.
He never had an official position, actually, during his phase 2 of his mycological life. But he was in Gainesville for almost 30 years. And during that time, he described over 700 species of fleshy fungi.
Okay.
Most of them were boletes or agaric. So lots of russulas, lots of boletes, lots of ammonitas,
Oh, is
things
that
like
the
that.
That's the type that you were kind of saying?
That's the one that Scott was talking about.
Yeah.
He was deciphering one of Merle's types.
Got it.
The one I was working on is from someone else, also from that era.
Okay.
So described from the 20s. His Gainesville period was, very end of the late 20s, but 30s and 40s he was just describing like a madman, and he didn't drive. So almost all of his collections are from, walking distance or places he could get a ride from the central part of campus. At that time, this right here was, just sort of the backwater behind Lake Alice. So this was actually, we're standing sort of right where he was doing a lot of his collecting. Before all these roads and buildings were here. And so a lot of the type localities for things are actually what is now campus.
Okay.
And so he was very active right here in Gainesville. Most of his material is from Gainesville, which is nice because when we collect something and it matches one of his species, and we can go back to these specimens, check that it actually matches, and then we have something collected near the type locality. So that's quite helpful sometimes.
Nice.
The downside, you referred to the humidity. So the downside
Yes.
is he was collecting during a time when there was not air conditioning in this collection. There's no real dehumidification. So his era of mushrooms were things that were dried at high heat and then put into a room that got as humid as it is outside and then got as cold as it was probably outside during the winter time.
Yep.
And so they were humidified, dried, cold, hot. And so we've had some very big challenges with trying to get DNA data from his collections. We're still trying, and we've made some progress using some methods that we're working on developing, but it's hard.
Yeah.
It's
That would be a
not.
challenge.
It's very time consuming compared to some other collections from further north, which seem to have weathered the conditions a lot better because they never got as hot or as humid.
Yep.
Yeah.
Got you.
And also just other fungi growing on them has been a problem, and insects too in the
That's
past.
a good point. Like, how would you decipher, if you're like, oh, this is part of that mushroom, but then like there's other fungus growing on there?
Yeah. So you basically need to use specific methods for the particular groups that you're working on.
Okay.
And there's definitely been at least one heartbreak where you get the packet out and you're like, I'm going to check that mushroom, and it's just piles of insect frass. We don't have a lot of those. None of them since I've been here.
Yeah.
But historically, that's been a big problem in a collection in the border of the tropics. So there are some challenges with Florida's environment. It's part of why we have so many mushrooms in the first place.
Yeah.
But it's not great for preservation.
Right. How many species or genera would you say is kind of collected in here?
I should know the
Rough
answer to that. I definitely have the answer somewhere.
Okay.
We have about 50,000 specimens.
Okay.
And the other thing that I could tell you is that when I first got here, none of this was digitized. So,
Yes, yeah.
in the first week of my job, someone came by and they wanted to know what fungi we had from Haiti. And it was like, I have no idea. We would have to go through every single
Like,
cabinet.
Good luck.
Right? Yeah, go for it. But I have no idea. But now the vast majority of our collection is digitized, so it's in a database.
Right.
So if someone said, what things do you have from Haiti? We could type it in, come up with a giant printout of anything. And it might take us a while, but we could find them and pull them out for someone who wanted to study them.
Yeah, that would be a cool idea for like a map. Like you could like see it a map wise and like all the
Yes.
different dots around the world.
Yeah, so we have some of those
Okay.
data available, and you can use some of the tools to map sort
Yeah,
of what we
some
have.
GPS and
Of
all.
course, the vast majority of what we have is from Florida, for reasons because Merle was a massive collecting machine, and he described a lot of things. But we do have things from other places as well. Yeah.
Neat. Wow. Yeah. There's a lot of cabinets here.
Yep.
What's-
It's a big collection for sure.
What's the oldest specimen that you have here?
Ooh, I also know that. So, later 1800s, we have some specimens. Many of those are actually exocati. So, exocati are basically what people used to do.
Okay.
I study fungi in a particular area. I have all these species I've identified, and some of them I've described. And basically what people would do is they'd make little packets, put it in a book, and send splits off to their friends. So basically you get a copy, and it's all little bits of the tissue that are put in sort of envelopes. In the early days, that was a lot of how, because there weren't really good photos. Printing was really expensive,
Right.
like all various reasons. And so you would actually send like a whole book with specimens. So those are called exocati.
Okay.
In some places at the herbarium or fungarium, they basically have them still as a book. But often in smaller collections, they would take them all out of the book and distribute them inside of the collection. So we look through, sometimes we're like, oh yeah, this one's from 1870 and it's from this exocati. So we have some of those.
Wow.
We have some early plant disease specimens that are, pre-1900. When I gave a talk about the fungarium pretty early in my time here, when I was still trying to learn about the collection, I found it was so interesting that almost all the collecting in Florida, almost all the earliest collecting was from northerners, people from New England and Virginia and things like that, that would come down, but only in the winter time. And I started doing a little more digging of like, well, what's going on? Why is it, all the early collections, they're only in the winter? And then it struck me. It's like you could get malaria and yellow fever in the summertime, and so none of those people would come. They would come in the winter. Mosquito season was over.
Yeah.
They would take a little steamboat up the St. Johns River or whatever, and they would collect in some of the towns like Mount Dora and these places that were, there were small towns there and little hotels, and they would collect right around those places.
Cool.
And then as soon as we started understanding mosquitoes and mosquito-borne illness and air conditioning and all that, then we have, material from all times of the year. But Florida was a pretty small place in the late 1800s, and so we don't have a lot of collections pre-1900 really.
That makes sense. One of the questions was, have you ever had a time where the Fungarium came in so much clutch, like you, you're like, oh, there's maybe something that we want to re-look into, and you're like, thank goodness we saved this specimen. Has there ever been a time, or maybe in the future? That
Yeah,
it could be
I guess the thing is, I've been training in mycology since 2000, so 25 years of doing this. So there's so many times where specimens have been really important, and I was lucky to have mentors that relatively early said, you need to save specimens and you need to organize them, and it's important for a bunch of reasons, and I got on board early.
Yes.
In the first couple years when I was a grad student, when I didn't have someone mentoring me and kind of explaining how to do that and why it was important, I guess the opposite is really the case. That there were times where I was like, man, I did not do a good job of saving that thing, and turns out that would have been really interesting.
Yeah,
so
exactly.
So I would say I've learned through bad experience early in my career, and now I'm like a hoarder.
Good.
But trying to turn it into a library, it's like you're naturally a hoarder, but you try to organize yourself enough that you can have the packets line up, and most of the material you need is in it, and the data are on the packet, and so but yeah, I would say there are many times where questions come up because things were not saved, and then you can't really answer some questions without a specimen.
Yeah, right, absolutely. It's better to keep it organized. It's not really considered a hoarder if it's all
if it's all organized. There's
a
a borderline between hoarder and an organized person. The one thing I will say is, so by comparison, let's say, for birding, people take photos of birds, and the diversity of North American birds outside of Mexico is not so high. So you could, just by photos, you can mostly tell all the birds apart, and so therefore, just having photos of the macroscopic structure of the bird you can answer a lot of the questions just from videos or photos. And that's true also for some plants, where like you can use Seek and it will tell you the answer because there's only so many plants, and maybe it can say, well, it's one of these 5 things.
Right.
But for fungi, we really need the specimens actually even more because a lot of times you need to measure the spores, and even then you might not be totally sure.
Right, the LBMs.
The LBMs, oh my goodness. And they hold a lot of really interesting information, but you really need a microscope or DNA or both to really dig in. So I think that's why we're here.
Exactly.
Yeah, and I think probably will become more important over time.
I was going to say, probably in the future, I think I was watching this news thing where they've collected all these squirrels from like decades, and now they have grown in size based on like them eating our food, and so maybe we can like go back in time to look at the fungi and maybe map the diversity or how it's grown, shrunk. Some mushrooms are now invasive, so there's just so many things we don't know until we've collected all of that to keep track of it.
So definitely fungi are useful for a bunch of other ecological and evolutionary questions. Here, this is a good one. Because this is a type, I believe. I should know whether it's a type. It's mentioned in the.
Oh, is that the Titans? Yes.
Yeah, this is a good one. So.
Big one that like you can.
Well, we can do it here. Is the light okay for you?
Yeah.
Okay.
Oh, I can smell it.
Yeah. So this is our oversized cabinet, but it has this example specimen of what was named as Tricholoma titans. So this is a good one for several reasons. Well, first, it's easy to see the specimen, and there's not very many things that are that big. So, it's kind of a specific thing. Also, you can see if you look in there, there's evidence of some damage from the past, which is a little bit sad. But but you know, they're soft-bodied organisms.
Yeah.
It's hard to keep them intact forever.
Right.
The good thing is we know what this species is really well now, so it's not hard. One thing that's really interesting is Dr. Kimbrough. He was the person, he got here in 1966, and then he retired in the late 2000s, and he described this as Tricholoma titans. But this species was not ever described by Merle because he never saw it. And when Kimbrough found it, it was only near Tampa. And it appeared to be new, like it showed up there and no one had seen it. So we don't know actually if it was introduced or if it was always in South Florida, but then just slowly has made its way further north with warming. Yeah. This species traditionally was only south of Tampa. Now it's pretty common in Gainesville, and it's made it as far north as Durham, North Carolina. They've had some collections, certainly in Georgia and South Carolina now. Wow. So I think that's an interesting one because we have pretty good evidence that over time its distribution has changed. Whether that's because of warming or people moving it, or, you know, there's always going to be some speculation. Yeah. But it's one where having these historical records, Merle collected lots of fungi here for a long time. He would have certainly described something that, you know, gets that big. Yes. Kimbrough was here, but didn't ever find that mushroom here, only in Tampa, and now we know it's moved north. So this is a good example for several reasons of why that is useful to kind of have these historical reference points for you.
So that's kind of how mushrooms are kept in just like plastic bags after they're dehydrated?
So that's a good question. If you go to some collections, they would frown upon using plastic bags. But the problem is it's Florida. And so my rationale has been, if there's ever a hurricane and this room has to be without a dehumidifier for 3 days, I would rather have things be in plastic bags, even though it's an imperfect vehicle for keeping the fungi. And so it's sort of a practical choice.
That would be so sad, like a hurricane just destroys the whole Fungarium.
Yeah, which is part of why we send material, like often important specimens, we'll send a split to the National Collection, to the New York Botanical Garden, University of Michigan. There are several big collections that have a much larger staff, sort of dedicated to collections all the time. Okay. And so we will often send our most important things, we'll send some small pieces to other places as sort of a backup. Because most of those other places are not in the potential, you know, category 3 or 4 hurricane path. So we try to keep that in mind. Yeah, so this is a good example of another taxonomic one. So this one is, well, it used to be called Latiparus persicinus. Now it's called, depending on who you talk to, Cusagoporia persicinus. There's a competing genus name for this, actually. But I guess the more important thing is that it was named in the genus Latiparus, which is chicken of the woods mushroom, and it has some things in common, but it doesn't really fit well. Okay. And it turns out that it's not very closely related with the Latiparus, like, and it kind of doesn't look like it either. Yeah, so I had a student who worked on that group and changed the name. So.
And then is that like a little sample taken out there? Or?
That was a sample that someone took out for microscopy and culturing. Yeah. Cool. As part of that research. So. That's cool. Just an example.
Good. I'm glad I asked about it. That's cool. Yeah, what was the last sample that you kind of sent up to look at?
Great question. Well, there have been several recently. So in my lab, we've had several different people working on truffle-like fungi that are describing totally new taxa. And so for those especially, we make sure that we send, if there's enough material, we try to send it to multiple places so that they will have them. A lot of times for our truffles, we send them to Oregon State because that's one of the sort of places where people have worked on truffles. So there are a lot of truffle collections there. And also New York Botanical Garden has a lot of truffles, so we'll send them there as well. So yeah, several different papers on truffles. And on some of those, they were actually describing taxa from South America. Okay. So we also always send material back to those. Cool. We have collaborators, and so they obviously want to have some of the material as well, which is appropriate, obviously, because those things were described from there. In the old days, people didn't do that. They would just keep it, and they didn't send it back to the place where it came from. So we work with a lot of local collaborators in Chile and Argentina. And so And a lot of times we only have half of it to begin with
Gotcha.
because they kept it. So.
Yeah, I guess that was another question was like, have you come across a species where you're like, oh my God, I've been looking for this collection for so long and I finally got it?
I would say yes, definitely. There was a species that we're working on describing that I collected in 2019 when I was in this relatively threatened forest in Chile.
Okay.
And we went back again, and I was thinking, man, what are the chances? But I really want to collect that again because it was only known based on one specimen.
Right, okay.
And I found it again. I was like, sometimes it works, you
Yeah.
know? Many times that when I've tried to do that, it doesn't work. So
Right.
every once in a while, you get a win.
I like that.
That's also one of the challenges of studying fungi, of course, is that they show up and then they're gone. If you're not there that day looking in the right place at the right
Right.
time, then you won't find them. And some of them are really legitimately rare things. I mean, like they really aren't very common, you know,
Right.
and then you combine not being very common with having to be there at the exact right day.
The factors or all of the things have to work together.
Yeah. So slime molds are interesting because, they're not fungi.
Yeah.
But nonetheless, they're in here.
Oh, cute.
So there was a Dr. Aldrich, Henry Aldrich, was here in the microbiology department for a long time, and he really specialized on slime molds, and he used them especially. He was also an expert in electron microscopy back when that was kind of a new thing, and so he used them as subjects for his electron microscopy. So we have a lot of his collections. They are not related to plants or fungi very closely, but they're here because no one else will take them, right? Like
that's where we belong. With the
yeah,
fungi.
exactly. So so they're different in several fundamental ways. Like they ingest their food. They actually do phagocytosis, which is much more similar in many ways to animals. So they sort of engulf their food. So they don't make hyphae, right? There's a lot of things that are clearly non-fungal, but because they make spores and they're kind of close to microscopic, or some of them are microscopic, some of them are close to microscopic, they've been treated with the fungi always. Yeah.
I was like, how do you preserve that? Like, how do you dry that out the same way that you
Well, yeah, I can show you. You basically just dry them,
just in cute little.
and then you glue them down. So you sort of, here I can hold it like that. You glue them down to the box itself.
Yeah, just little black bits. That's cool.
Yeah. So this is not a particularly important research collection in terms of people don't usually ask to borrow them the way they do the fungi.
Right. Hobby collection. So we're here with Tyler Hacking, and I just did an in-person interview touring a Fungarium in Gainesville. But you had this really cool topic. I think you're starting your own Fungarium?
My second one, actually.
Okay.
So I've started one in a university, Utah Valley University, in their herbarium, and I was formerly trained by the taxonomist and herbarium director, and learned a lot of different preservation techniques. But the thing with universities is that they have limited access. Technically, the herbarium is often a laboratory. It's a secured room. You need a card to get in. You have to get on a list. It's not open to the public. So donations, specimens from the public, that can be really difficult, right?
Okay.
That whole process, it's almost just the biologists are donating.
Yeah, very secluded
Yeah.
and restricted in a sense.
It's a limited data set.
Yeah, because so when I did my interview, it was through the university, and so we got the cool tour of the Fungarium.
Yeah.
But yeah, you had that really cool input of like, why do you think Fungariums should be more open to the public versus university?
I think that it should be merged better, right? Science should be more open access. The problem in herbaria and taxonomy is that curation protocols are hyper specific and vary, and so you literally have to get a degree like in taxonomy to be, and herbaria creation and things like that to be able to do that.
Right.
So they're highly specialized and particular, or it doesn't work and you can contaminate things and destroy things. So I understand why there's a separation, but I've taught lots of people. I've taught so many people. So what we're doing in the Mushroom Society of Utah, I'm the chair of their science committee, and they gave me the power to do what I want. So I'm like, we're making a public Fungarium. It's going to be open access. So for now, it's compartmentalized, which means that different members of the Mushroom Society are taking on different genera of interest. So I've taken on Morchella. I have the entire Morchella collection. They're in different containers. I have archival boxes. So we do have a budget, and I spend that on archival containers, things that don't leach, that allow us to, we use desiccant packets of different kinds, silica beads to control humidity, and just practicing those things with people is a really great way to learn how to do preservation.
It's more people involved.
And then we do it with, at the same time, with iNaturalist, and then if you do it properly, you can send those for sequencing, and then that contributes to science.
Okay.
And the public can do that.
Yeah.
Absolutely.
Is there a downside, though, to making it public versus, more private?
Yeah, because if the public doesn't use the right protocols, then it can contaminate the specimen. But that's all practice. It's a part of learning. That's what the science students do as well.
Yeah, okay.
So we mess up while we learn how to do it correctly.
Yeah, and during this whole Telluride Mushroom Festival that we were just at, you were always there to, blend the two, the academic and, the public, community.
Divide.
Yeah, so why do you think that's important to really blend, those two communities in a sense?
First of all, it limits science. Definitely. Not just in a data context, but in a... Here's the one that gets me, okay? I think the most important mycologist in the world is Meredith Blackwell, and she contributed to ITS, and that's fundamental. It created entire new fields of science.
Wow.
Excuse me.
So why the divide?
Yes.
Why the divide? Yeah. So here at the Telluride Festival, I met maybe 5 PhD biologists, right? That's ridiculous. There's so many.
Yeah.
And they're busy people. Professors are the busiest people in the whole world.
True.
So going to public events can be difficult, but it would be really nice to see a lot more academics at the public events. And by the way, MSA, Mycological Society of America Research Conference in Portland, Oregon, 2026, next year.
We're going.
It's open to the public. You can go and see all of the research that every single undergrad and postdoc student is doing in the United States of America, and professors present on their entire lab's research. Last time I went, we learned about the psychoactive porcini, the Dentinger lab in Utah is doing. I met the inventor of mycelium bacon, Dr. Meredith Blackwell, who contributed to the development of ITS sequencing for the molecular genetic analysis of fungi. So really, really important, high contributing, high impact mycologists who are just so busy in the lab doing science that they don't come to the events. And so it's logistics, but I'm one of the few people that walks both worlds, and I think it's important because we need to bridge that gap. It makes science function better. It makes education better.
Yeah, well, thanks for having that input. That was really eye-opening.
Yeah.
Where can people find you and get a hold of you?
Plug in. Okay. drgaia.com, Dr-Gaia is my website, but you can find me just Tyler Hacking. I'm on a lot of socials, and I'm sure I'll be in the description below.
Yeah, and also you're starting a podcast too. Want to tell people what that is?
Yeah, I just interviewed you as my first episode.
Yeah.
It's called Path of the Scientist, and it's all about what caused people to start the path of science, what they're doing on that path now, and where it's leading them. So if you'd like to be interviewed and you're a scientist, hit me up. I don't care if you have a degree or not. If you contribute to science, you're a scientist.
Awesome. Well,
These are the leaves, or in some case flowers, that have been sort of expanded to become galls.
Yeah, that's really cool. New species possibility.
Yeah, well, it's on a species of Leonia that's only on Dominican Republic.
Okay.
Hispaniola. So there's a species that's shared between Haiti and Dominican Republic. And so we found a lot of that Exobasidium on it. And it looks a lot like the ones on Leonia here, but we think it's likely a new species. We're still figuring that out. Yeah, so, we can pull out some pickled things to look at.
Look at that Anita egg.
Wow.
That's fun.
There's a Sebasina. That's kind of a cool one. It's a jelly fungus. Yeah, we got lion's mane. We got some Romarias there.
Oh, that's the one. Okay.
That's Romaria. Yeah,
I was
sorry.
the coral type of.
This is good. You were talking about morels earlier. We've got a pickled morel up here. Some pickled morel. There
Looks
we
like
go.
somebody drank half the juice.
I know. Well, some of them are not sealed as well as they could be.
So-
This is a cool one, Winnia. We don't have this one in Florida, but this one is really cool. It's a mycoparasite.
Oh,
It
nice.
attacks armillaria.
Okay.
Attacks another fungus.
Wow.
That's kind of neat. Yeah, so
Yeah, wait, can I get
you got all
some
yeah.
of the lion's mane here?
Totally.
That's cool. And the shaggy mane type of.
Got all
This is
kinds
the
of
drunken
good
mushrooms
stuff.
I was talking about earlier.
They are, yeah. They're the drunken mushrooms. That's true.
Wow.
These are props for our class. These are all fungi. This part, right? These are all like fungi food products. Got a little bit of snake skin in here as well. So that we use for baiting for chytrid fungi. So we have all kinds of white fungus. Little tidbits in
Is that the
here.
kind that eats fingernails?
Thanks again for watching Flora Funga Podcast deep dive into what a fungarium is and more about the lab research aspect. It's starting to rain, so hit that subscribe button. Have a great week, my scientists, and go learn something new today. Peaches!